Thanks, Matt. My name is Abdulman Malsali. I'm from the Department of Pathology at the University of Otago in New Zealand. And actually, about 2 weeks ago, I just submitted my PhD thesis. So thank you. Thank you. So I will be telling you today one part of my PhD project, which is about the use of ABC.RAP to identify novel methylation biomarkers for childhood leukemia. But before I start, let's define leukemia. What is acute leukemia? So in normal bone marrow, the hematopoietic stem cells differentiate into myeloid and lymphoid lineage. And the lymphoid lineage differentiate into T cells, B cells, and natural killer cells. So in leukemia, there's a proliferation of the immature lymphoblasts that occupy the space of the bone marrow and compromise the functions and differentiations of the other cell lineages. So patients with leukemia tend to present with anemia, bleeding, and risk of infections. Now, it's widely accepted that epigenetic has a role in cancer development, and one of the epigenetic modifications is DNA methylation. DNA methylation is the addition of methyl group to the cytosine molecules that are followed by guanine. And so here a sequence of gene and has CG sequences that are highlighted in bold. So we often refer to these as CPG. So P refers to the phosphate group that's in the backbone of the DNA. So if I represent these CPG sites by lollipops and the rest of the sequence as lines, then I could represent them in the image that you can see at the bottom. But addition methyl groups to DNA molecules, what does that mean biologically? It's not changing the sequence of the DNA, right? So what's the role of methylation in terms of cancer? Well, in cancer, in some of the regions of the gene where the CPG sites are located very closely to the gene expression region, we often call this region as the promoter region. It has a role in the transcription regulation of the gene. So if the CPG sites on the promoter region are unmethylated, then there's no prevention for the transcription machinery, so the gene is switched on, so the gene will be transcribed. However, in cancer, and some of the genes that are involved in cancer, like tumor suppressor genes, the CPG sites that are in the promoter regions are densely methylated, so they trigger a signal like a stop sign for the transcription machinery to switch the gene off. There are a number of methods that were used to measure the DNA methylation on the human genome, and one of these methods is the use of 450K array system. So 450K array is an array-based system that contains more than 450,000 probes, and each probe interrogates a CPG site. There are a number of analysis tools available for the analysis of 450K array data. Unfortunately, the majority of these tools focus on identifying differentially methylated genes based on having one CPG site that's differentially methylated. For biologists like me, I find that it may lead to weak validation. So what I'm interested in is finding genes that contain multiple differentially methylated CPG sites, not just one. And in order to achieve that, I set a criteria for the selection of the methylation biomarkers for leukemia. And the ideal tool that I want to use is a tool that can arrange the 450K array probes for the gene of interest in a way that's similar to the genome browser, so I can dissect the gene into different genomic locations, like the promoter and the gene body, and which is similar to the genome browser as I mentioned. And also the ideal tool could also plot the methylation values for the genes of interest for leukemia samples and controls at the same time. And based on that, I could select a region that has a high difference in DNA methylation between leukemia cases and controls. For my genes that I'm interested in, I want to have genes that have at least more than 50% methylation difference between leukemia cases and controls. So before I moved to ABC.RAP, I searched the literature to find tools that can help me with that analysis. Unfortunately, there is nothing available that helped me to meet these criteria. So I developed a pipeline to analyze the 450K array data. And before I talk about ABC.RAP, I would like to share with you my experience with R. In fact, at the beginning of 2016, I had no idea how to use R. Not even the simple functions like how to open files in R. But at that time, I joined an R workshop. I started to learn some basic skills, and I applied that to my data. And with the encouragement and support from my supervisors, Professor Ian Morrison and Dr. Rob Weeks, and the incredible support from the R community, I was able to transform from an Excel user to an R user. And I was able to build a pipeline that I think it will be useful for the community as well. So here's my contribution to epigenetics community and for our community. ABC.RAP has 3 stages: the processing, analysis, and results. So the package doesn't really deal with normalization and batch effect adjustments, but it filters out the probes that are non-specific, and they were known to align to non-specific regions in the human genome. The analysis part has 2 parts. There's the global DNA methylation analysis to give you an overview about the methylation status of leukemia and controls. And there's the differential methylation analysis, which applies students' t-test and delta-beta analysis to identify CpG sites that are differentially methylated. In the results section, it provides a list of genes that are differentially methylated, and these genes have at least 2 CpG sites that are differentially methylated. And also, it plots all of the probes for these candidate genes to select a region of interest. So the typical output of ABC.RAP is a plot like this, where you could see the methylation values for leukemia and control, and leukemia samples here are represented by red circles, and controls are represented by blue triangles. And you can see that the methylation is ranging from 0 to 1 on the y-axis. At the same time, there's also the package provides a plot that calculates the difference in DNA methylation, which is the cases minus controls, and also provides a plot for the distribution of DNA methylation for the cases and controls. And based on these plots, you could select a region for target sequencing or for validation. Also accompanying with these plots are results tables, and the region that was selected in the plots are highlighted in red here. So in order to find my methylation biomarkers, I went to the data repositories and searched for publicly available 450K array data. And I found 7 data sets that were suitable for the analysis. I applied stringent cutoff values, so I used a Bonferroni correction on the student's t-test, and also I decided that I want genes that have at least 50% difference in DNA methylation. Out of these 7 data sets, 4 of them provided lists of differentially methylated genes. So I took these lists and tried to combine them in order to find genes that are consistently methylated across different data sets. So when I combined the list from 4 cohorts, I found 5 genes. And then when I combined the list from 3 cohorts, I found 36 genes. Then I went and inspected these 36 genes to find regions that I can select for my sequencing assay. Here's an example for one of these genes, KCNA4. And you can see that the methylation pattern for this gene in B-cell leukemia and T-cell leukemia is quite remarkable. The pattern is highly similar between these 2 different data sets. And based on that, I selected regions for target assay. So my validation experiments included blood samples from 17 healthy adults, 13 healthy neonates, and also blood samples from B-cell and T-cell leukemia samples. And I designed primers for the 11 target genes that I selected from my analysis. And I used a two-step PCR approach using MySeq for sequencing. Now, this is just a brief overview about the two-step PCR approach that I used. So the first PCR reaction is to amplify the target region with the gene-specific primers. And the second PCR reaction is just to add the Illumina adapters into the amplified regions for the amplicons to hybridize on the Illumina flow cells. So then the samples were combined or pooled and then sequenced on the MySeq. So to help the interpretation of the results, let's go back to the lollipops. So if I change these lollipops into rectangles and put them next to each other, then they will form a line. And so the methylated and unmethylated lines represent methylated and unmethylated genes inside cells. Then if I stack these lines on top of each other to represent the sequence reads, then I could have an image about what's the phenotype of the cells in the sample. To analyze our MySeq data, we use Galaxy platform to process the raw files, and then we use the BioQ Analyzer HT to align and analyze the methylation values. Here's a typical output from the BioQ Analyzer HT. And you can see that the sequence reads are arranged in rows and the CpG sites are arranged in columns. And by looking at the methylation pattern for these sequence reads, you could see that they are different, suggesting that these methylated reads are coming from different type of cells rather than BCR amplification bias. So let's have a look at some of the results. TES, which is one of the tumor suppressor genes, you could see that it's densely methylated in leukemia, while adult blood samples and cord blood samples didn't show any methylated reads. That's remarkable. Another example is an NEFM, shows a low level of methylation in adult blood samples, but the majority of cord blood samples were unmethylated compared to a very dense methylation in B-cell and T-cell leukemia. Let's have a look at the rest of the samples. Let's have an overview about the results. So these are my adult blood samples. And as you can see, so I started to squeeze the methylation results for each of these samples into a very small rectangle and put them next to each other. And you can see that TES is completely unmethylated in all of these adult blood samples. What about the other genes? There's a very low level of methylation in adult blood samples for some of these genes. Cord blood samples, they are completely unmethylated. The remarkable thing that all of the B-cell leukemia samples were densely methylated. And there are a little bit of variation in terms of the methylation level in the T-cell leukemia samples. To compare our results, the sequence results, with the 450K array data, I extracted the methylation values for the CpG sites that correspond to the 450K array probes, and I tried to compare the results with the 450K array methylation data. So I plotted them next to each other, and as you can see, the controls here are represented by blue color, while the leukemia samples are represented by red and green colors. The difference between controls and leukemia samples in the MySeq data is quite remarkable. It's hugely different compared to the 450K array data. This suggests that there is a really good validation for this gene, and this picture is actually a representative for all of the 11 genes. Majority of the scientists, when they validate their results, they barely have a really good validation. In my case, I'm getting really good validation, and that's because of the ABC.RAP, the concept that was used in ABC.RAP to identify and select these genes was quite stringent. So with that, I would like to thank my supervisors, Professor Ian Morrison and Rob Weeks, and the wonderful people in Morrison Group, and also the funding bodies. Thank you. Well, the promoter region is not really well defined, but we use the 1500 bases before the transcription start site as a definition. Yeah.
Did you have any control genes which are not thought to be influenced or be important in leukemia to see if they will also increase methylation?
So the question is there any control genes that are not involved in leukemia and they are influenced by methylation?
Or increase in leukemia in your samples?
Yeah. So test gene, which is the tumor suppressor gene, the first gene that I showed you, there was a paper that was published on it, and it was tested by my supervisor, Rob Weeks, and they did bisulfite sequencing, they did COPRA and other type of tests, and found to be densely methylated in leukemia. The other genes that I found are novel genes. They were not reported before in the literature. Well, I can't really comment whether other people will be able to reproduce these results, but I have a really high confidence that they are methylated in leukemia. Does that answer your question?
It was more whether there were genes thought to be unrelated.
Yeah.
That may, so is it a specific effect on these tumor suppressor genes, or in leukemia you just get lots more methylation?
So you're referring to like gene ontology analysis, right? Or is it like a...
I'm just having control genes that are like these, so just genes that we don't think have anything to do with tumours, but just to make sure that it's not just an effect that in leukemia, because of cell turnover, you just get more methylation throughout the genome, rather than being specific for tumour suppressor genes. It's more like a negative control.
Yeah. Well, so the genes that I have, they are not really tumor suppressor genes. Yeah. So NEFM and other genes, it's only TEST that was known to be a tumor suppressor gene. And the idea from choosing these genes is because we want to use them as methylation biomarkers. We are not really interested about whether they are involved in the pathogenesis of the disease or not. We just want them to use them as a marker for other purposes in the project.
If there are no further questions, let's congratulate Abdul for submitting thesis.
Thank you.
And thank all the speakers.